Hello everyone, welcome to Biophilia, your all-time study. Today we are going to start a new chapter about biotechnology and its application. This chapter would be helpful for you once you have understood the previous one, that is biotechnology principles and processes. If you haven't, then please visit the previous video. The link will be provided in the description box. Enjoy watching this video ahead. So if you like this video, then don't forget to like, share and subscribe, and do hit the bell icon so that you get the notification for all the upcoming videos. So just a little flashback going to understand how the biotechnology was coming into action. So there was a round development in all the fields. They made fashion, might be medicine, or it might be anything that you imagine. हर जगह पे डेवलपमेंट हो रहा था in the past century. Many huge impacts were produced. और जैसे जैसे डेवलपमेंट हो रहा था, it was creating all the luxuries, all the benefits, so that the population could have a better facility. So since it was all available to the people, the population growth happened. The population growth rate had a big spike, and because of the big spike there was a sudden growth of number of people. और जब लोग बढ़ गए उनको खाने की ज़रूरत पड़ी, and the basic need of food also become less. People became increasing, population growth बढ़ते गया, but the food quantity remained the same because food industry में कुछ change नहीं हुआ. It was the same as it was, but it needs to be changed. People बढ़ रहे हैं. Even during that time, India had been independent, so many people were coming back into India. People were starving, famine, everything happened at all same time. But it was the time when scientists have to do and think about how to improve the quality and the quantity of the food. So with that, the application of biotechnology started. So during that time, not suddenly the biotechnology emerged, but people started with the three options. सबसे पहले people thought about to use the agrochemical based engineering. So agrochemical means it was the procedure where it was been using large amount of pesticides which was not actually told to be used, but during that time it was the most suitable time to increase the production. Production increase करने के लिए pesticides use किए गए because pests were the problem that were decreasing the range of things that were produced. So insecticides were used, insects were killed, herbicides were killed, fungicides were killed, everything was used to maximize the production. But during what thing, all these chemicals that were used in huge quantities, कम समय के लिए ही सही, but the production increased, but that was not the permanent cure. Whatever chemicals had been used, now it had been started to accumulate into the groundwater level का जो सारा पानी था, that all started to degrade and get infected with these pesticides chemicals. सारे plants का जो toxicity था वो हो चुका था. It was all on saturation. Now whatever use of pesticides, insecticides, fertilizers, nothing was working because now it had come to a constant rate. पहले प्रोडक्शन इधर था, सडनली इट हैड अ स्पाइक एंड नाउ इट वाज ऑल ऑन कांस्टेंट. अब ये कांस्टेंट लेवल को चेंज करने के लिए पीपल अगेन स्टार्टेड यूजिंग समथिंग एल्स. एंड नाउ एट दिस टाइम व्हाट डिड दे हैव? दे हैड द सेकंड कैटेगरी, that was organic culture. People started to tell that instead of using the chemicals, let's go back to the place where we already were, that is use of organic fertilizers. So organic farming started, which was with the use of manures, the biofertilizers, biopesticides, biocontrol, everything related to natural ecosystem. नेचुरल इकोसिस्टम पे आना वापस जरूरी था because land conditions खराब हो रही थी. Land was getting polluted, and now that particular land was becoming infertile and no crop were getting cropped. So it was very important to save the land, and to save the land and keep the production on it, organic agriculture had become the medium. But organic culture को करने के लिए it cost a lot. it takes time, and even the production at that time was becoming less. So still people had to work with it to come back and to save the soil so that they could have at least some amount of growth. But again, it was the time that it needed a drastic change, and that drastic change has been provided by the genetically engineered crop based agriculture, जहाँ पे क्रॉप से अब उम्मीद छूट चुकी थी. एक्सटर्नली वट एवर यू कुड प्रोवाइड, यू हैव गिवन ऑल द बेस्ट मटीरियल, बट अगर उसके जीन्स में ही, उसके खून में ही there is nothing, then how can you improve the production? So at this time, instead of changing the external things, why don't we change the internal of that particular plant? And then the start of genetically engineered crops begin. So at this time, the great person that was Norman Borlaug had gone, wherein he started the use of Green Revolution. Green Revolution was the period in which there was a significant increase in the agricultural productivity. But again, Green Revolution lasted for a short period of time, but that gave us the period to undergo the genetically engineered crops. So Green Revolution में एक पूरा सक्सेस नहीं था. It wasn't a complete success because of four major reasons. First of all, it has been using all the chemicals. Agrochemicals जो थे developing countries में, they were very expensive. Cost बहुत ज़्यादा था. And because of that particular cost, the production cost increased, and now the selling cost increased. That made impossible for the localites to buy those. That wasn't a use, क्योंकि अगर बहुत ज़्यादा rate है, people won't buy, then what is the use of the production? So that wasn't a success. Second major problem was whatever the increase in yield was there, it wasn't being able to increase more and more with the conventional breeding. एक प्लांट को दूसरे प्लांट से क्रॉस करने पे नई ब्रीड बना के it wasn't giving a very huge hike in the yield. Now it needed something that could make its production. So starting में इतना था, conventional breeding से इतना आया, but now it needed a big spike. This big spike was produced by the genetically engineered crops. This was with the conventional crossing, और ये था जब you did nothing, right? So this big spike was needed, and for that big spike, now you cannot lend upon the huge amount of fertilizers. You cannot have, because of fertilizers, जो pollution हो रहा था, the harmful effects could have been there. And to reduce the harmful effects, the use of fertilizers and chemicals needed to be minimized. So again, production was decreased. So all because of this reason, Green Revolution was a boon for some period of time, but later on it wasn't useful. So it wasn't a complete cure, not a complete success. The next part we see that जो भी problems हमने discuss किए, the four problems. Now how could you resolve those? Because problems को solutions देना जरूरी है. So the solution had become the genetically modified crops. Now you need to change something in the internal composition so that you can increase the production. So for that reason, genetically modification could be done in two types. The first was addition. Add something that you want in that particular plant, or you remove something that you don't want. उसी के वजह से modification होगा, and that might help you to increase the production. So gene addition was done with addition of more new genes, and subtraction was to eliminate or to inactivate one or more plant genes which are already existing in it. So these kind of plants, say for example, majorly यहाँ पे food problems थे, so plants were used. So once it was successful on the plants, then bacteria, fungus, animals, everywhere it was manipulated in the same way. जैसे यहाँ पे किया gene addition subtraction in the plants, that was carried out in bacteria, fungi, and animals, and all these organisms combined together, which were manipulated, altered using this, was called as genetically modified organisms or the transgenic organisms. Now, the examples of these kind of transgenic or the modified organisms. So we are seeing here majorly with the respect of agriculture. So agriculture may one of the major changes that was done using genetics, that was the golden rice. golden rice is an example of gene addition to be remembered. Gene हमने add किया है that we wanted. So what gene did we add? We wanted to fight against vitamin A deficiency. During the time vitamin A deficiency की वजह से many people were turning blind. So eating just that rice, staple food of rice and fish in the southern country, it wasn't useful. People were developing vitamin A deficiency, and you cannot change the staple diet of the population. So now you wanted something so that it could be used by the people as their staple diet. Still, it will provide the vitamin A. So now, how could it be done? So instead of changing the staple food, let us change the composition of the staple food and accumulate vitamin A into that particular rice itself. So then there was the born of transgenic variety of rice which had good quantities of provitamin A. What was the provitamin A? It was the beta carotene. Beta carotene was the principal source of vitamin A because in animals you can convert beta carotene into vitamin A. So beta carotene अगर rice में है, once you eat the rice digested, beta carotene can be used by your body to produce vitamin A. So rice खा के you are achieving the vitamin A concentration that you need, and because of the beta carotene inside the rice, which was genetically added into it, which was manipulated by human intervention, the white rice that you usually saw, now it had turned yellow in color, and because of the beta carotene it was yellow. The name of the rice was kept as golden rice. So just eating rice, if it could solve the vitamin A deficiency, that was a great success in the field of genetically modified plants. Now, the second example of gene addition was the plants which were allowed to make their own insecticides. So, own insecticides, because people were not allowing the use of external chemical insecticide pesticides, but that wasn't the case. Insecticide needs to be used because insect used to come and destroy the crop produce. So, there is a technique कि अगर आप समझ सकते हो कि अगर आपको प्रोडक्शन इंक्रीज करना है, if you want to increase your produce, instead of increasing the production more and more, because production will come to a stagnant place at some time, but what you can do is you can reduce the losses. अगर losses reduce होंगे, production automatically increase होगा. So using this particular method, the next gene addition procedure was used. The principle about this next topic is the Bacillus thuringiensis. This is a bacteria which used to produce endotoxin, and this endotoxin when used to be entering into the insect, अगर किसी इंसेक्ट ने या किसी और चीज ने बैसिलस thuringiensis को खा लिया, then this endotoxin used to be cleaved by the protease enzyme because it was a protein toxin. So proteases used to degrade it, और ये toxic activity दिखाता था under alkaline conditions, and in the mid cut of the insect that used to damage the epithelium को damage करके pores होते थे because the toxin used to destruct the epithelial surface, and because of the pores there used to be cell swelling, cell used to die, and that used to cause a great pain to the insect stomach, and that would lead not to eat anything, और insect अगर कुछ खाएगा नहीं तो it used to starve to death. So that was the Bacillus thuringiensis mechanism to save itself so that the insects or any other thing should not come and eat that bacteria. So that was a self-defense which was observed, and it was an idea to produce own insecticides in the plants. कि अगर ये endotoxin insect को मार सकता है, then why not use this endotoxin inside the plants so that the insects would die and not eat the plant rather. So the endotoxin now wanted to go in the plant cell. How it would go? Originally ये bacteria में था. Now you wanted to go in the plant cell. How would you do it? So it is very important to know कि since the toxin was a protein base, प्रोटीन था, प्रोटीन कैसे बना था? प्रोटीन was made from the RNA. Okay, RNA came from where? came from the DNA, and from where came from the gene, because particular gene is coding for the protein. So if you want the protein, you need the gene. So now gene needs to be separated. अब gene को separate करके अगर आपने plant cell में डाल दिया, then that gene is now going to produce that protein inside the plant. And that was a great thing. We could isolate this gene, we could identify it, and we can transport it to the plant cell. Now how can you isolate, how can you manipulate this gene into the new plant cell? That was all in the previous chapter. So previous chapter, that is the biotechnology principle and procedures, the link is provided down in the description box. If you don't remember or you want to know more about it, how it is done, तो आप प्रीवियस चैप्टर को डिटेल में पढ़ सकते हो, एंड दैट वुड हेल्प यू टू नो दैट हाउ दैट जीन इज एक्चुअली बीन प्रोड्यूस्ड इनटू द न्यू प्लांट सेल. सो नाउ once the gene is into the plant cell, एक plant cell के अंदर डालने पे, as the plant cell will divide, the entire plant is going to produce that endotoxin. Now once that endotoxin is produced by the plant, now it is whenever any insect comes and tries to eat that plant, endotoxin will enter into the plant body and the insect will starve to death. So that was the idea behind the plants that create their own insecticides. Now own insecticides creating plants को बोला गया BT crops. What they were called? They were called the BT crops. Now what crops were made into BT crops? It was the cotton. After cotton there was corn. It was also carried out in brinjal. It was carried out in soybean, also in tomato. Right, it was all done in here for the BT crop production. And the major success for BT crops was in cotton, क्योंकि cotton को आप खाओगे नहीं, you're not going to eat, it was not for human consumption. So nowhere the endotoxin was going to come in contact in the human consumption. So BT cottons in crop was very much popular, whereas the others were also being used, but majority it was successful in cotton. So BT cotton, cotton का क्या use होने वाला था? Cotton का fiber use होने वाला था. So the fiber is also now having the toxin in it. Any cotton bollworm is trying to come and eat and feed on the fiber, क्योंकि जो fiber है वो fiber किससे बना है? Fiber is made up of cellulose. It is energy rich and the insect wants to eat the cellulose to gain energy. Now the fiber should be protected because the fiber is nothing but equivalent to the price that the farmer is going to get. So fiber needs to be protected, and that protection will be provided by the endotoxin that is being provided by the transport of the gene. A gene कौन सा था? Gene was the cry gene. Now why it was named cry gene? You might be wondering. So it was considered as कि जो toxin था वो insect के body में जाके destruction करता था, that used to create a lot of pain in the insect used to cry. So why it used to cry? Because of the endotoxin that was produced by the gene, so the name cry gene. So this cry gene, once it was added onto the plants, it was effective against some particular insects coming from Leptodopterans, that is tobacco bollworm or armyworm, or from Coleopterans, that was the beetles, and the Dipterans, that was flies and mosquitoes. So against these, the endotoxin was successful. And even one more thing about कि जो cotton may, once we have seen, so cotton may there is a worm that is cotton bollworm. So cotton bollworm के लिए specific gene was used, that was the cry1AC and cry2AB, right? cry1AC and cry2AB genes were used, whereas for the cotton, corn में क्या था? Cotton borers थे. What it was? Cotton had borers. So cotton borers used to have cry1AB gene, right? So using these genes, the cotton borers, corn borers, and the cotton bollworms had been eliminated and the produce was saved. So that was about the plants that used to produce their own insecticides. The next is the gene subtraction. अभी तक जो देखा that was gene addition. हमने कुछ add किया जो हमें चाहिए था. Now is the time we subtract something that we don't desire or we don't want. So that was wherein the use of antisense RNA came into action. What is antisense RNA and where it was used? It was used in pest resistant plants. हमने insect resistant plants के that was using gene addition. Now pest resistant using antisense RNA. So here there was, instead of a bacteria that was Bacillus thuringiensis, here we have the main hero, that is the nematode. And this nematode was the inspiration क्योंकि ये जो nematode है, this used to infect the tobacco plants. Tobacco plant में ये roots में जाके settle होता था, and once it used to enter the roots of the tobacco plant, it used to cause nodules, and this nodules used to cause decreased absorption of water and minerals, and because of decreased absorption of water and minerals, the plant used to die or used to reduce the production. And you know that tobacco is a very important as in the drug industries. So for that reason, it was very important to increase the production, or at least not to, get infected with this nematode. So what used to be done, strategy ऐसी यूज की गई थी कि ये जो निमेटोड है, हाउ इज इट कॉजिंग द नोडुल फॉर्मेशन. तो नोडुल फॉर्मेशन हो रहा था बिकॉज ऑफ अ स्पेसिफिक आरएनए. नाउ दिस आरएनए इज जनरली अ सिंगल स्ट्रैंडेड आरएनए. Now, single-stranded RNA, once it used to produce protein, because of that specific protein, it used to cause nodules. अगर आपने RNA से प्रोटीन बनने के स्टेप को ही रोक दिया, what will happen? No protein production, no nodule formation. So that was the technique which was used. So now to stop the RNA producing protein, you need to stop the translation process. Now again, here the words that how is RNA converted to protein, you need to see the videos about how the mechanism of translation occurs. So once the translation has been stopped, now when it will stop? When the single-stranded RNA is converted into a double-stranded RNA. अगर आपने उसको single strand से double strand बना दिया, then you can stop the protein formation. So the same thing was applied here as well. So now it was the RNA interference which was used to silence the genes. यानी जो RNA था उसको silence करेगा. It won't allow it to function, and it won't allow to function यानी protein formation will be stopped. So now how is that extra RNA being transported? So here comes the biotechnology part. So again, using the vector that is from the agrobacterium, now the RNA was supplied to the host cell. So अभी tobacco cell के अंदर क्या रखा है हमने? Tobacco cell के अंदर we have transported the RNA. Now say for example, this is the tobacco cell. Okay, tobacco plant cell है, and inside the tobacco plant cell you have inserted a single stranded RNA, okay, using agarobacter vectors. Now once that nematode tries to enter into the plant cell, okay, समझो ये हमारी nematode आई, and nematode now is trying to enter into the tobacco cell to form nodules. Now that should not happen. अब तो जो nematode है, nematode के पास भी एक RNA है. This RNA is going to be complementary to the RNA which is found in the tobacco plant cell. Now what will happen? The plant cell is having one RNA, that is the red colored one, and one that is from the nematode. Now what will happen? There will be pairing of it, और जैसे ही pair हो जाएगा of the RNA, it will be converted into a double-stranded RNA. And now this double-stranded RNA cannot produce the protein. And if it is not producing the protein, now the plant cell has been saved from the infection. So this same thing was done, and because of that, once the double-stranded RNA was formed, now the plant cell does not entertain the double stranded RNA. So double stranded RNA was ultimately broken down, and once it was broken down, now the plant was saved from the nematodes यानी pest resistant plants produce हो चुके थे. So by this way you can understand how the use of entering of this single stranded RNA was important. अब ये single strand को add करना, that was just in one line, but how it was added was all taught in the previous chapter. So you should know the basis of how it was added, how it was identified, all that thing. Now why it was done, how it was done, that is all clear about the gene subtraction. Now we see the biotechnology in application of medicine field. हमने agriculture में देखा how to increase the production, how to save it from the insects, how to save it from the pests. Everything was seen. Now in medicine field, medical field में how it was useful. So first of all, what were being used? So there were recombinant therapeutics, hormones, enzymes, or antibiotic vaccines. Many things were produced using biotechnology. So first of all, what was the advantage? That it was utilized in medicine field as well. First of all, it was enabling mass production because herein the cells were used majorly as bacteria जिनका multiplication rate बहुत ज़्यादा था. So because of that, mass production was possible in small amount of space. It was more safe and more effective as well, and it was not having any induced immunological response. It was not hampering, it was not affecting adversely onto the users. Now, how it was the users were getting affected before this biotechnology, let us see in the next example. So the next example was about the recombinant insulin. So recombinant insulin बनने के पहले, what was the scenario? You should know that. So recombinant insulin के पहले, still that there were patients which were suffering from diabetes mellitus. Diabetes mellitus में there were pancreatic cells which were not able to produce insulin. So now it was the time to produce artificial insulin. Artificial insulin previously was being obtained from either slaughtered pigs. Slaughtered pigs के pancreas से it was extracted and the insulin was given artificially to the diabetes mellitus patients. Now, what was happening because of the slaughtered pig? It was the pig, not the human, but it was similar kind of insulin because of which some people used to have great results, but 1 in 100, 2 in 100 used to develop allergies. What used to happen? They used to develop allergy. Now, allergy should not occur, but to stop this allergic reactions, now what was done? It was the time to change this slaughtered pig pancreas insulin production. Okay, pancreas में से pig के pancreas में से how much insulin can you obtain? You can obtain 1 vial, 2 vials, but the demand was increasing. Pig slaughtering was being increasing, which was not valid. So again, insulin production was decreasing, demand was increasing, cost increased. So to reduce the cost, it was the time to have a change, and this change was nothing but the recombinant insulin. So instead of using pigs for production of insulin, now it was the time that the bacteria will start producing insulin. Pigs, पहले तो उनको खाना खिलाओ and then you slaughter them. So again the pig जो खाना खिलाओगे पिग को, that is very costly. Now the pig becomes costly. Slaughtering के बाद the cost increases for insulin. But bacteria, what do bacteria needs? They don't need any food in large quantities. आपने उनको सिर्फ थोड़ा सा nutrient medium में डाल दिया. Bacteria are happy to grow there in very less amount of cost. So because of that, bacteria low cost maintenance, कम जगह चाहिए, production ज़्यादा है. Recombinant insulin was a great advantage at that time. So it was again another thing, making of insulin. It was necessary so that कि insulin जो भी बनाएंगे, that insulin cannot be administered orally. आप उसको oral dose में नहीं दे सकते हो. Now why can't you give it orally? Because as you might know that insulin is a protein. Okay, it is a protein based enzyme, and since it is a protein based hormone, now what it will do, it will be attacked by the protease पेट में alimentary canal में, since it is a protein based hormone, protein will get degraded. अगर वो पेट में जाएगा, small intestine में जाएगा, protein को break करेगा, insulin is of no use. So before absorption, insulin is gone out of the body. So it was very important that it was given intravenously. उसको produce करना था to give it directly into the blood supply, not to enter into the alimentary canal. So for that production of insulin, insulin was a protein that was a boon. और कोई hormone क्यों नहीं बनाया? Because insulin being a protein, and as you know very well कि अगर आपको protein है, you should be very happy in biotechnology, because protein comes from RNA, RNA comes from DNA, and you have a great technology to administer this DNA, change it any way that you want. So protein अगर आपका desired product है, be happy, be thankful, you have genetic engineering that can change everything. So DNA now was to be located, that is the gene needs to be located. So for the first time it was identified कि जो gene चाहिए for the prohormone of insulin, it was on the chromosome number 11 of the human being, क्योंकि human insulin form करना था. So human chromosome number 11 पे the gene of production of insulin was obtained. Once it was obtained in the year 1983, Eli Lilly, an American company, started to isolate this gene, and with the gene sequence, the DNA sequence, there was production of two chains of this insulin. So using the plasmid, that is PBR322, it was being located in the E. coli bacteria. E. coli को बढ़ाते गए बहुत सारी colonies बढ़ाई, and the transformed one was useful by the selection of blue-white screening, because beta-galactosidase was used. Now what is beta-galactosidase? What is blue-white screening? How is it done? Everything has been discussed in the previous one, so that you can see and understand how the blue-white screening is done. So with this, two chains बनाए गए, A and B. Now what is this A and B chains? So for that you need to understand the structure of insulin protein. So insulin protein एक छोटा सा protein है which is made up of 51 amino acids. This 51 amino acids are taken into two strands, that is chain A and chain B. The chain A is going to be made up of 21 amino acids, and the chain B is going to be made up of the remaining 30 amino acids. Right? So combining the insulin of 51 amino acids is produced. Now ये chain A or chain B को, how will you link them? It will be linked with the disulfide bonds. So disulfide bonds will keep the chain A and chain B together. That will be forming the insulin. Now, insulin जो बनाया गया, जो originally ऐसा होना चाहिए था, but once you are making it artificially, there is some change. और वो change क्या आया? The change was the addition of chain C. Chain C was additionally added, and because of the chain C, it was now called as proinsulin. So proinsulin has the chain C, but now the chain C will be broken down, and then there will be the insulin formation. So here you can see the red colored is going to be your peptide B, and the blue colored one is peptide A, and based on that there is the gray colored, that is the C chain. C chain को free peptide बाहर निकाल दिया गया after the end of the production, and then chain A and chain B will be joined together by the disulfide bonds. And this was the first creation of artificial insulin by using DNA recombinant technology, and then it was termed and it was named as humulin. What it was named? It was named humulin because it was human insulin, and instead of using pig insulin, now it was the same human insulin that the patient was not able to produce. So that was the very great advantage in the medicine field, decreasing the cost of insulin, and the demand was also being met by this production. The next we see is the gene therapy. Gene therapy was a different kind of clinical diagnosis के बाद एक treatment का जरिया था कि जो genetic disorders है, जेनेटिक डिसऑर्डर्स जहाँ पे जीन्स के डिफेक्टिव होने की वजह से द पेशेंट यूज्ड टू सफर. नाउ इट वाज टाइम टू से गुड बाय to certain diseases which were caused by this genes. Now you could change those genes, you could add something so that the missing product is formed, or you can delete some mutated ones. So for that reason, the first clinical gene therapy was given in 1990 to a 4-year-old girl. और ये 4-year-old girl किससे सफर कर रही थी? She was suffering from adenosine diaminase deficiency, that is ADA deficiency. Now this adenosine diaminase is an enzyme. Now you know that enzymes are what? The enzymes are nothing but proteins, and you should be happy in genetics, that is biotechnology, if it is a protein hai, again and again repeating, it is RNA coming from the DNA. So once you have the DNA, you have the gene, you can change it the way you want. So now the adenosine diaminase deficiency could be cured because of the addition of the gene. So अब क्या होगा कि अगर ये अमीनोसिन डायअमाइनेज की डेफिशिएंसी वैसे की वैसे रहने दी, समझो बिफोर द बायोटेक्नोलॉजी people were born with this deficiency, but what used to happen? Because of this deficiency, there was absence of lymphocyte proliferation. Lymphocytes produce नहीं होते थे T cells के बिना. The ADA deficient children used to be invaded by many bacteria, many viruses, जिसकी वजह से severe recombinant immune deficiency disorder हो जाता था, that is SCID. So SCID was, that means entire immune system was crashed, no immunity, and it was all the small little kid was having no protection. It was being invaded by each and every bacteria. हर कोई आके infection produce करता था, death हो सकती थी. And now the treatment part came because of the biotechnology. Now people can go and have a gene therapy जहाँ पे 3 methods use किए जा सकते हैं. First, in the starting, once the child is adult, kind of 4 years, 3 years का है, 5 years का है, at that time you can have a bone marrow transplantation. So bone marrow transplantation या enzyme replacement therapy हो सकती है, but it wasn't a complete cure. वो आपको बार बार करना पड़ेगा. And since it is the adult one, उसके पूरी body form हो चुकी है, so it is not possible for the entire bone marrow to get replaced, so it is not functionally possible. सेकेंड हो सकता है ट्रीटमेंट कि अगर आपको थोड़ा और पहले पता चला है, इन द इन्फेंट स्टेजेस, यू कैन हैव द इंट्रोडक्शन इट इन द lymphocytes. Lymphocytes में अगर इंट्रोड्यूस किया, what will happen? Since lymphocytes will multiply again and again and again, comparatively a lot more amount of the deficiency can be cured, but again repeated infusions are needed. Many more sessions will be kept with the infant so that the lymphocytes, all the lymphocytes, having the functional ADA gene. And now last and the third most important one that could be done with the modern diagnosis techniques, कि अगर आपको पहले से पता चल गया है कि the embryo or the fetus that is growing inside the womb is having the ADA gene that is deficient, then what could be done? The embryonic stages में ही, जब वो छोटा सा है, it is not even born, it is still dividing to form the fetus. At that time, in the embryonic stages, if you are adding this gene, what will happen? अगर embryonic stages में ही if you have added the gene, it will one gets divided into multiple cells से जब पूरी body बनेगी, the entire body will be filled by the gene. You don't need to transfuse again and again because each cell is going to have the gene that you have infused in the embryonic stages. So that was a great success. for the babies who used to get their infusions in the embryonic stages. So that is same thing that has been shown to here in the diagram कि जो bacteria था that was carrying the human ADA gene. Now how did you form this particular bacteria? That is in the previous chapter. Previous chapter may we have already seen how to create the recombinant DNA and transfer it to the host. And once it was transported into the host, that is the baby, then the gene therapy was successful. So by this way you can understand that how biotechnology, that is एक जीन को host cell के अंदर डालने तक, how can you use this and give different kind of remedies, maybe it in the field of agriculture or maybe it in the field of medicine in humans. Now the next thing was the modern techniques. ADA deficiency है, ये अगर आपको पहले पता चल जाए तो आप उसको प्रिवेंट कर सकते हो. Similarly, there are many other more diseases that can be prevented by the modern techniques of diagnosis. अगर आपको लेट डिटेक्ट होता है कैंसर, अगर आपको स्टेज 4 पे जाके डिटेक्ट होता है, then what can you do? The ultimate stage is death. But if you can have a method जहाँ पे आप स्टेज 1 पे ही डिटेक्ट कर लो, you can have better life. So that is the stage wherein you need better techniques for diagnosis at early stages. So there are three major modern techniques that one can use. The first is PCR, wherein you can have the low concentration of bacteria of viruses जिसके symptoms अभी तक दिखना शुरू भी नहीं हुआ है. Just एक हाथ दो बैक्टीरिया है, एक हाथ दो वायरस पार्टिकल है, not एक हाथ दो, just like minimum quantity. And even with that minimum quantity, if you amplify that quantity, अगर आपके body में sample में if there is 100 molecules that are not visible, but if you amplify that 100 से अगर आपने 10 billion कर दिया है because the use of PCR, then what can be done? It can be diagnosed at the early stages. So अगर infection है, इन्फेक्शन में 100 सेल्स को मारना इजी है या अगर आपके पास 1,00,000 सेल्स है उनको मारना इजी है. इट इज मच इजियर टू कंट्रोल द invasion of 100 particles instead of 1,00,000 particles. So जब 100 पे है आपको normal technique से पता भी नहीं चलेगा कि this person is suffering from something. Until and unless it reaches 1,00,000 तब तक आपको symptoms दिखेंगे. अगर आपको symptoms दिखेंगे the cure will be difficult. But in this case 100 है, symptoms नहीं है, cure will be easy. Then why not go and target the 100 part? 100 पे cure easy है, जल्दी है, symptoms नहीं है, but still we have the PCR that can help you. The second thing can be the use of ELISA. ELISA जहाँ पे you can use the presence of antigens or you can see it by detecting the antibodies. So for example, if you are using the technique of tuberculosis, for example tuberculin test होता है wherein you can use the ELISA जहाँ पे you can add the antibody, जहाँ पे आपको antibodies पता है. Suppose in the sample you have some sort of antibody that is known to you. If you add the antigens, that can tell you कि already this person was infected previously and the body has produced immunity against it. Now you can do opposite. If you have an unknown sample, say for example कोई viral bacteria है आपको, you don't know that there is unknown bacteria in the sample. Okay, unknown bacteria in sample, you have known antibodies, unknown antigen. There you can use ELISA and find out अगर कलर चेंजेस होते हैं. सो हियर यू कैन सी स्टार्टिंग में इट इज कलरलेस, एट द एंड प्रोडक्ट यू हैव अ पेल येलो कलर. अगर कलर चेंज होता है that means it is positive for the unknown cases. It might be unknown antigen, it might be unknown antibody, but with the use of ELISA you can detect it in small quantities, the presence of antigens or the antibodies. The third is the autoradiography. Autoradiography में you can use the single stranded DNA or the RNA which will be tagged by a radioactive material. Autoradiography, radiography radioactive material को उसके साथ attach किया जाएगा and that attached particle will be called as a probe. So single stranded DNA and the radioactive together called as a probe. Now this probe will be given into the clone cells. Clone cells में जब भी ये divide करेंगे, that would be considered and a radio autography will be produced. Now this radio autography will be seen onto a film, and once you can see on the film, what can you be observed? फोटोग्राफिक फिल्म पे म्यूटेटेड जीन्स नहीं आएंगे और जो नॉन म्यूटेटेड है वो दिखेंगे. So based on that, whatever is visible and whatever is not visible onto the film, based on that you can identify whether the infection is there or the genes have been mutated या कोई deficiency है. All that can be signals at the earlier stages. So that is the modern techniques of diagnosis. The next is the use of transgenic animals. हमने अभी तक जो भी transformations किए थे, that was majorly using the bacteria or the microscopic ones. What about if we use it in the multicellular ones like the animals? So transgenic animals बनाए गए which were used in 3 important fields. One that was medicine, second that was agriculture, and the third that was the industrial applications, that was the manufacturing part. So medicine में transgenic animals कहाँ कहाँ use हुए? So the uses of these transgenic animals in the field of medicine, it was done in the first to understand about the normal physiology and the development. क्योंकि transgenic animals, say for example, if you are studying about some kind of a disease, say for example, if you want to see how does the cancer stages grow, or you want to see how does the diabetes mellitus work, or you want to see any other disease, how is the effect onto the animal, all that can be studied. So जैसे diseased condition study किया, वैसे normal physiology can also be studied in the transgenic animals. The third important one is the biological products. Biological products means what was done is there was a production of alpha-1 trypsin, alpha-1 antitrypsin. This is the product that was being used, and alpha-1 antitrypsin was used for curing the emphysema. Emphysema which used to cause lung disease. Emphysema is a lung disease. Lung disease को prevent करने के लिए alpha-1 antitrypsin was used, and that was created because of the transgenic animals. Even there was a formation of first transgenic cow. Now this is an important question, that is first transgenic cow जिसका नाम था Rosie. She was named Rosie. जैसे first cloned animal कौन सा था? First cloned animal was Dolly. वैसे Dolly the sheep. The first transgenic was a cow which was named Rosie, and she was used to produce alpha lactalbumin. What she was used for? She was being used for production of alpha lactalbumin, which was at the rate of 2.4 grams per liter. So whatever milk she used to produce, the alpha lactalbumin was produced at the rate of 2.4 grams per liter. Now that was the transgenic cow that is used in medicine. Now vaccine safety, wherein the transgenic mice are used. So transgenic mice क्यों use करते हैं? Transgenic mice because once the vaccine needs to be available for the human, it needs to be undergoing the influence कि animals को कुछ नहीं हो रहा है, then humans won't be affected and it is safe to use that vaccine. So transgenic mice को use किया गया identify the effects of polio vaccine, और polio vaccine जब transgenic mice पे use किया गया, then it was allowed to use it on humans. So again transgenic mice was formed because of the use of biotechnology. And finally the chemical safety testing is also being one कि drugs जो है, any new drug needs to be tested on the lab animals, and these lab animals, अब ड्रग्स जो बनाए हैं, ड्रग्स किस पे बनाए जाते हैं? ड्रग्स आर मेड ऑन टू सम काइंड ऑफ डिसीज. अब ये डिसीज नेचुरली तो ऑकरिंग नहीं है, okay, it is not naturally occurring in that animal. So how will you create that disease? Create in the animal with help of this transgenic animals. and that animals would be then used for the chemical testing of the new produced drugs. Then the next was in the agriculture field जहाँ पे breeding use होना था. So plant breeding, animal breeding, the transgenic animals were used. Then for the quality production as well. So it was important कि quality में there was more production of milk. Milk ज़्यादा से ज़्यादा produce हो because demand ज़्यादा है. But it was also one important demand that was to have less lactose. Now the question arises that why less lactose? Lactose कम करके क्या होना था? Milk production increase किया, it was accepted. But why less lactose? There were two major reasons. First of all, अगर lactose कम होगा, then it is easy to digest. Okay, it will be easy to digest. It won't be heavy. Major second reason was it was for the lactose intolerant people. जिनको lactose से allergy है, those people can now drink milk because lactose won't be available in that milk, and such kind of animals were created with the help of transgenic production. And the last one was the disease resistance, again in agriculture field. And the last one was industrial applications, जहाँ पे the Nexia Biotechnologies in Canada, they had been using this biotechnology to introduce the gene into the lactating goats. Lactating goats में उन्होंने ऐसा gene add किया that was coming from the spider. अब स्पाइडर का जो भी सिल्क था, द स्पाइडर कॉब बनाने के लिए जो भी प्रोटीन यूज कर रहा था, that protein used to be so tight, it used to be so flexible enough, and it should not break. So spider का जो silk thread होता है, that is very flexible, it is very stiff, and that can be used for many purposes. So अगर ये स्पाइडर जो है ये lactating goats बनाने लगेंगे, so along with milk the goats also started producing tiny silk strands. So along with milk these strands were used for producing bulletproof materials या for heavy duty ropes या कुछ भी जो बहुत ज़्यादा यूज होने वाला wear and tear कम करने के लिए, flexibility बढ़ाने के लिए this spider cobweb was used, and that was produced by not the spider but the lactating goats. So after all these things हमने अभी देखा कि स्पाइडर का जीन गोट में डाल दिया और ह्यूमन का जो जीन था वो हमने बैक्टीरिया में डाल दिया, and we started producing that was not exactly related with the nature. We started interfering the nature, and for that reason it was very important to have some rules and regulations कि हम कुछ भी कहीं पे भी use ना करें and we not violate the nature rules. So for that reason there was some ethical issues, and to suppress those ethical issues the Indian government has set up an organization that is Genetic Engineering Approval Committee. So any sort of biotechnology changes that you want to make, any genetic engineering you have to produce, it needs to be approved by the GEAC. If it is approved, if it is been giving the permission, then and then only you can follow up with your procedures. So why is it necessary? Because once you are using the animals in biotechnology, there is a lot of suffering to them. आप उनके nature के खिलाफ जा रहे हो. Once you are creating the transgenic mice for the study of diseases, आप एक healthy mice को infect कर रहे हो to produce the disease and understand the effects of those disease on the mice. So won't the mice be suffering from it? So mice को suffering हो रही है, it is getting पेनफुल कंडीशन है, हो सकता है माइस की डेथ हो जाए इफ योर ड्रग्स डू नॉट वर्क, हो सकता है कि इंसान मरने से पहले कितने सारे जानवर मर गए. So for all those animals to have some protection against them as well, so that the human cannot make anything and test it out on animals, the committee needs to give the approval. Second important thing was once the animal are used in production, for example the cow that was the transgenic cow Rosie, she was used to produce milk that was having the lactose thing, या फिर गोट थी जिसने सिल्क बनाया फ्रॉम द स्पाइडर. सो वंस यू आर यूजिंग इन प्रोडक्शन, हो सकता है ह्यूमन को लालच आ जाए लालच आने की वजह से प्रोडक्शन इंक्रीज करने के लिए इट इज एक्सप्लॉइटिंग द एनिमल्स. It is not understanding the status of the animal as a living being, but it is using it as a production unit. So, for reducing that kind of a stress to the animal, the status of the factory needs to be shifted to a status of a living being. वो भी एक जिंदा जानवर है, वो जानवर को समझ होनी चाहिए. It should not be exploited in any way for the genetic engineering. So, for that all reasons, the committee needs to provide you the approval for your further proceedings. The third important is once you are changing any genetic makeup of the animal or the bacteria or any species, अगर आपने कुछ और introduce किया है, it is transferred to the next progeny. Now what will happen once it is getting transformed from one progeny to other? It is violating the integrity of the species. Integrity of the species damage हो रही है. That should not occur. Species should not vanish because nature balance should not affect. So again, that is the third important point that needs to be taken care of regarding the ethical issues. Fourth point is once you are transferring any sort of human genes in the animals, जैसे human insulin बनाने के लिए human insulin from the chromosome number 11 was shifted onto the bacteria. So it might be the bacteria, it might be other animal for some other production. So you are diluting the concept. Okay, so जो human genes है वो आप animals में डाल रहे हो, animals से आप काम करवा के you are using those. So the transport of humanness is decreasing and that should not occur. You should also respect those animals which are being used for those procedures. Biotechnology is being disrespectful for the living beings, and it is, you are making everything in the respect for the better utilization of human beings. It should not happen. It will just exploit others and benefit the humans. आपने cow लिया, goat लिया, mice लिया, bacteria, anything, any living being is just using, you are using those organisms so that you can benefit the human population, but you are not anywhere using it for the benefit of the same organism or some other organism other than the humans. So that is again a big problem, that you are disrespectful to the other living beings. You just care about the humans. That should not occur. So to control those feelings, the committee needs to be set up so that it will have a check on what procedures have been taking place. The last one is the biodiversity may pose unforeseen to the public risk. हो सकता है कोई ऐसा एक्सपेरिमेंट करने की कोशिश करे because of which instead of being boon for the human life as well, हो सकता है वो environment को खतरा हो, या हो सकता है वो पूरी public को ही destroy कर दे. It might happen that it might be a biological weapon that is being produced. So all that things needs to be taken care. And once all these things are understood, if all these things are okay with the committee, then and then only you are being provided a legal uh, permission to carry out your further genetic engineering. Now the last is about the patent. Patent is nothing but going to be having three criterias. Three criterias. Patent कब मिलता है? You have heard this word patent. I got a patent for this invention or for this. Now, on what criteria does a person get a patent? Does every product do it get a patent? No. First of all, it needs to have some novelty. What is novelty? Novelty means the innovation must be new. आप ऐसा नहीं बोल सकते हो कि someone has created a fridge, same fridge अलग color में बना दिया and you say I want a patent. No, the invention, the innovation should be new. It should be not been made previously. That is patented. Second is going to be, it is non-obviousness. It should be known to other people, but it should not be documented. Means people should understand what has been happening over there. And the third thing is utility. If you are making that product, if you are giving the patent, is it useful for the humans? If it is useful, then the patent will be granted. If it is not, then why to give a patent? If it is not useful, then you won't get a patent, even if you successfully pass the first two criteria. If it is not useful, you won't get a patent. The third thing is किस चीज़ों पे आपको पेटेंट मिल सकता है, for what things can you actually apply for a patent. So you can apply it for an invention or a product. You can have it in the form of an improvement which was already be done. आप उसको improve करोगे, अगर जैसे for example nowadays you see that teaching fields there are different kinds of machinery. तो एक machinery है, एक fan है, वो फैन वैसे ही चलता है, बट इफ यू डू समथिंग न्यू इनटू दैट फैन then you can have a patent because you have improved the earlier invention. Third thing is process of generating a product. अगर आपने फैन बनाने के प्रोसीजर के अंदर कोई चेंज किया है, that can also grant you a patent. And finally a concept or a design जो अभी तक बनाया नहीं है, it is just a thought, but that thought, for example, the greatest patent is for with the Tetra Paks. Tetra Paks के पास patent है, and this patent is that they cannot use the Tetra Pak production for any other company. It is patented. No other can use this Tetra Paks under their own name. So that is one example of patent. The procedure, the product is everything patented. The idea is patented, and that is example for the patent that can help you to understand. Next is the diversity that we have regarding patents. So this is a question not exactly related to patents, but you need to know for the MCQ's point of view. So the richest diversity of rice is seen in India. Over 2 lakh varieties of rice have been seen in India alone. So that is a huge scope wherein you can apply for patents. And the next one is 27 documented varieties of basmatis have been grown in India. So rice 2 lakh hai, but basmatis are 27. So these are just values that you need to remember with the respect of MCQs. So MCQs ke liye both the things are important. The next we see is biopiracy. What is biopiracy? That is the use of bioresources by multinational companies. Very important word is multinational companies and other organizations without proper authorization from countries and people concerned without compensatory payment. For example, if there is a tribal area, right? एक tribal area है, in that tribal area there are many trees, many flowers, there are many more things that can be grown. For example, एक tree है, okay, on that tree there are some beautiful berries. अब ये beautiful berries जो है, that is all maintained by those tribal people, right? There are many tribal people which are living in that area. Those tribal people are maintaining that fruit. Okay, now for example, if some multinational company comes, okay, any multinational company, any MNC comes and tries to steal that fruit, saying that we have the right onto this fruit and now this fruit is ours. We are the people who can touch this fruit, no others can touch it. So entirely tribal people are forbidden to use that fruit. Entirely the fruit consumption should be given to the MNC. Now what will happen? जो भी growth है, जो भी चीजें हैं, जो tribal people को मिलना चाहिए, instead of giving it proper payment to the tribal people or whether any proper documentation key, we would have a sit together, we will have a chat. No, there is nothing as such. The entire fruit is taken away by the MNCs. That is biopiracy. So, to protect all such activities, the Indian Parliament has recently been producing a bill in the second amendment. This amendment was done in the Indian Patent Bill, and such issues wherein the use of patents or the biopiracy, all that things were considered so that it would give a better rights to the local people. हो सकता है कि जो indigenous चीजें हैं, for example, the 27 varieties of basmatis in India, they are originating, they are the rights of the Indian people. But what if they patent it in some other country, say America tries to patent it or any other country tries to patent it, then it is a big loss because all the money will be going to the MNC but not to the local area. So the big companies can hire each and everything, and then the local elites, the developing or the underdeveloped nations, would suffer because of the biopiracy. So biopiracy is going to remove the rights of the community, majorly the developing countries, because developed countries have their great financial support. So the private institutions, they can, have the rights of those people, and they can get the patents as well, because majorly the developed cities are the ones who are going to grant patents on an international level. So the loss is for the developing nations against the non-developing ones. So all these things needs to be taken care once you are understanding about the genetic engineering or the biotechnology. So the patents, the biopiracy, all these terms need to be cleared, and that will help you to have a successful creations of yours in the future. So that was all about to understand about biotechnology. In the previous part, we had seen about the principles and the processes. And in this part, we have seen how these processes are majorly used in the field of agriculture, in the field of medicine, in the formation of transgenic animals and whatnot. We have seen covered everything in detail. I hope this helps you, and we will come back soon with some other chapter that will be helpful for you in your exam. Till then, don't forget to like, share, and subscribe, and do hit the bell icon. Stay connected to Biophilia. Until next time we meet. Bye bye, everyone. Thank you.